So I'd like to now introduce Christian Ionescu-Zanetti, who is Chief Technology Officer and a co-founder at Fluxion Biosciences. And I will turn the presentation over to Christian to get started with the webinar.
Hello, everyone. Thanks again for joining our presentation. This is an exciting time for us at Fluxion. We're about to introduce our IsoFlux system for CTC separation and recovery. And this is really the first presentation where we talk about in full detail the operational principle of the system, as well as a lot of the results in terms of analyzing patient samples and going on from separation to a number of downstream analysis modalities. I'm going to start the presentation with a disclaimer. Our consumable kits as well as system are listed as a Class 1 device with the FDA for in vitro diagnostic use. The use listed is blood dilution and CTC sample enrichment. The CTCs can be taken on for further analysis, but the analysis methods that we'll talk about today are for research use only, and we will include full instructions as to the prescribed use with the packaging. So we set out to develop the system with a few key requirements in mind for serving the CTC market. The most important requirement in our mind was to be able to have high CTC recovery, meaning hitting a certain minimum threshold of cells recovered for a majority of the patient samples that are collected. We believe that's important in order to enable answers from a number of different downstream analysis modalities, and that's what we'll be talking about today. Another important feature that I'd like to point out, we opened up the use of different antibody cocktails. I think everybody appreciates that we need to get away from just using EpCAM to separate out circulating tumor cells, and I'll be talking a little bit about that. But essentially, we have a IgG linker that can be used with a number of different antibodies or combination of antibodies, and some of our collaborators have used this modality. A lot of the data you see today is using EpCAM as a separation antibody along with immunomagnetic separation. The CTCs, another key addition or another key piece of innovation that I'll be talking about today is the ability to simply transfer CTCs to downstream analysis and be able to recover a full number of cells from the separation consumable and be able to transfer viable cells for downstream. That enables a number of analysis modalities, and we'll be talking about DNA abnormalities, mRNA measurements, as well as protein. So just a brief overview of the various approaches that have been used to separate CTCs out of the bloodstream. As you know, there are efforts focused on using size and filtration to separate out CTCs. There is affinity panning, where sometimes size is used in conjunction with antibody binding. Cytometry is a way to do it based on immunostaining. We've chosen to take the approach of using immunomagnetic separation, but addressing some of the key shortcomings that that method has had in the past. In particular, some of the issues with this method have been the limited isolation efficiency. We have addressed that by combining immunomagnetic separation with a microfluidic environment where we achieve very exact control over the forces that the cells experience and are able to process the cells effectively in a microscale separation zone. So to summarize, the technology that I'll be talking about uses magnetic beads that are preferentially bound to CTCs. It's something that has, as a method, a lot of validation behind it. However, what we've added to the mix is a microfluidic flow environment, proprietary design that focuses the cells onto a small separation target. We call that the CellSpot technology. It's essentially a disk that accumulates all of the CTCs and allows for simple retrieval without any sample loss and transferring the CTCs to downstream analysis. So the system format, and by the way, you're seeing an image of the system here. You'll actually be able to see the system in flesh and blood at the World CTC conference that's coming up in Boston. But it's basically a benchtop system. It can operate either in or out of the hood. It's focused on a touchscreen interface, and it can run up to 4 samples in parallel. What we've used is pneumatic pumping to move fluids from one well to another of the consumable that you see on the right. What that allows us to do is to have no sample contact between the between the sample and the instrument. That's important to manage contamination. After processing the samples, which are 1 per cartridge inserted, the CTCs are automatically transferred to a microcentrifuge tube, and that's the sample format that the user sees at the conclusion of the experiment. So going into a little bit more detail, we've developed workflows that are focused on providing the sample in a viable format in a microfuge tube ready for molecular analysis. So there are basically 3 different phases to each experiment. There's a sample prep phase where magnetic beads get added to the Buffy coat. It takes about 30 minutes hand-on, and then there's a coupling step. The total time is about an hour. We then load the sample into the instrument, and this is the automated part where both the wash buffer and the sample are moved through the separation region. That takes at most 40 minutes, and then the tube is available for removal at the end of the experiment. So just zooming in a little bit on the separation region, and this is the heart of the system really, we have the immunomagnetically labeled CTCs as well as white blood cells traveling through a microfluidic channel. That's shown here left to right. And what happens is there's this microscale separation region where we have a removable cover and a permanent magnet. And the cells get, the positive target cells get drawn up towards the magnet, and they remain immobilized on what we call the cell spot. What confers advantages to this approach is the fact that we can very accurately control the balance of forces that each cell experiences. We have drag forces due to the fluid flow, as well as the magnetic forces that act on the beads. So because of that, we can achieve a very high isolation efficiency, as you'll see, and we can get a majority of the circulating tumor cells immobilized next to the magnet. So here's an alternate view of the same design. This is actually closer to reality in terms of what happens in the instrument. So what you have on the left here is the same step I showed before, the target cell capture step. And you can see here that the magnet holder is a permanent magnet, and its holder basically looks like the cap of a microfuge tube. And that's exactly how it acts. So this is what we call the cell spot. After the cell separation is done, we simply remove this, and all of the target cells are held in a hanging drop. that's essentially about 2 to 3 microliters right below the magnet. That means that our essential elution volume is very, very small, and that prevents a lot of issues associated with contamination that are experienced if a large amount of elution buffer needs to pass through the system. So we have this really small volume hanging drop, and then the cell spot becomes the cap of a microtube shown here in spot 3. And essentially, from there on, the user can just mix the cells with lysis buffer or template as needed for downstream analysis. So we focused on this, on keeping the volumes down and keeping the sample very concentrated, because we felt that was important for a successful downstream assay. So again, focusing in a little bit on the consumable, we have a sample inlet well here. This is where the conjugated buffer coat is added. We have a microfluidic flow channel, and the cells pass through what are called the isolation zone before heading on to the waste well. So this is how the separation happened, and 4 of these samples are processed in parallel. At the end of the experiment, the blue tube here gets removed, and it gets placed on top of the recovery tube, and the sample comes out of the instrument, and that's essentially what the user's presented with at the end of the experiment. Now, I've talked a lot about recovery. I'd like to start talking about how we validated that with a large number of clinical samples. This is work that we've largely done with a number of collaborators from either biopharma or CRO companies or clinical research investigators. So as you can see here, we've looked at a number of indications, and we've used 2 different modalities of obtaining the primary sample. One is whole blood. That's probably something you're used to. We're just starting with a draw tube of about 7 mils. We've also done a lot of work with apheresis-derived samples, and this is work that was done with in collaboration with Bayer. And we think this is a very interesting new way of obtaining the primary sample for CTC recovery, and it's yielded a lot of very nice results in terms of numbers of CTCs recovered and the numbers of patients presenting CTCs. So I appreciate you may not be able to read the slide carefully, but this upcoming slides are essentially a summary of the data you saw there. And what we focused on here is just being able to determine what % of the patients we'd be able to get an answer from. In other words, a big challenge, I think, in the field has been that because not enough CTCs are recovered from a high enough percentage of samples, one can't always determine things like mutational status for enough patients to make this a viable approach to a diagnostic. So what we've basically done here is we've binned the data and looked at what percentage of patients have, through the accepted counting methods, either more than 5 CTCs per sample or more than 10 CTCs per sample. The reason we've chosen these numbers, you'll see soon, is because a lot of the downstream analysis methodologies have a limit detection that's right around 5 CTCs. So we picked that number as a point where it was we would have a high probability of being able to complete the analysis successfully. So these are the recovery percentages from whole blood samples. As I told you, apheresis samples are very, very good at providing high recovery. Again, this work was done with Bayer. Initially, it was done as a sort of exploratory way of getting a lot of sample and being able to distribute samples to a number of different sites. I think that they have since perfected the technique to a point where they believe it will be usable in the clinic. So finally, part of that Bayer study was comparing samples processed at a number of different sites using the existing Veridex technology and comparing that to recovery on our system. And this is just a subsampling of that. That should give you an idea of just how much more efficient recovery can be by using this approach. And again, this is just a subsampling of 17 clinical samples here. And if you look at the statistics there, you have a lot larger numbers of cells. Usually, there's some really high CTC numbers for certain patient samples, which drives the average number of CTCs up. The median number is around 200 per sample for our platform as compared to about 8 for the CellSearch platform. Now, if you translate that into what percent of patients present greater than 5 CTCs, which we think is a very important metric, essentially in this data set, we get 100% of patients over that threshold, whereas their previous methods are right around 60%. So I also wanted to talk a little bit about the background, and we have a few matched samples where we looked at the total number of cells as a surrogate for background. So basically, this is going in and counting the number of nuclei. I'd like to point out that we were very careful about matching analysis methods here. These were processed through exactly the same analysis methodology in terms of imaging and processing those samples. It's using automated counting routines. So what you see is that while we also experience background, the background is reduced by a decent amount. And what you should expect for our samples is a purity of about 1 to 15%. Now, across all clinical samples that we've analyzed, the content of white blood cells per isolated sample is in the range of 500 to 3,000. That basically translates to about 100 to 400 white blood cells per mil. And this is how the data is presented here. Okay, so I'm going to talk a little bit about our analytical data, and this is sort of an obligatory type experiment where we show linearity of the recovery depending on spike-in number. So these are spike-in levels of either PC3 cells, a medium EpCAM expressor, or NDA cells, which are low EpCAM expressors, and those are plotted against the recovered numbers of CTCs. The important thing to remember here is whatever antigen you're using or combination of antigens for completing the separation, we wanted to be sure that even when the expression was low for those particular proteins, we were still able to recover the sample. And that's what this shows here, is that we can recover a majority of CTCs viable for both expression levels. All right, so in the next portion of the talk, I'm going to be focusing on the downstream analysis methods that we think are most promising in order to actually provide a readout of the state of the CTCs. And what I presented here is 3 different categories. Obviously, people like to look at a number of different characteristics of these cells, and they're all important. One is DNA aberrations. Next is gene expression as measured by mRNA content, and the other one is protein content. Now, a lot of these have a history in the clinic and a history of acting as a diagnostic. We've listed some of the clinically relevant companion diagnostics down in the bottom, and I think that the number of these types of drugs is going to grow over time. A lot of companies have a stated mandate to develop a companion diagnostic with all of their cancer drug development programs. So starting out with DNA analysis, we've worked with Life Technologies in a R&D collaboration in order to validate a real workflow in order to have a sensitive mutational assay that can take IsoFlux samples and perform whole genome amplification and then go down and look at individual mutations in a panel of, in this case, it's mutations of the KRAS gene. And we've really worked with them in concert and have been able to develop this to a point where the sensitivity limits are well established. And I'm going to show you a little bit of that data. So here's an example of just starting out with gDNA equivalence. This is really the first step in validating this type of assay, where we dilute out MDA cell DNA equivalents after DNA isolation and go to lower and lower cell numbers in order to see where the cutoff is in sensitivity. So basically here we are plotting a delta CT of the mutant assay minus the reference assay. And the two lines that you see, the green line is the positive control. It's what you obtain in terms of delta CT if only MDA cells are used without any background. The wild type gDNA control is up here, and you'll see that the points at 0 cell equivalents are all above this delta CT cutoff line. So essentially that's how the cutoff is established. In this case, it's about a delta CT of 21. So what you should be able to see here is that even for really low cell number in a high background, and by the way, we used a background of 10,000 cell equivalents just in order to test this under a worst case scenario. So we've taken 10,000 sort of white blood cell equivalents in the background, and here you see the number of cell spike-ins. So we've come up with about 4 as the cutoff for gDNA equivalents. And then we moved on to spike-in samples. So here what we're plotting is the number of MDA cells spiked into a whole blood tube that go into then the downstream analysis. So we typically will take a number of cells, spike them in, and then perform the isolation and divide the sample up. So what you see here is that the same type of sensitivity limit that we determined using gDNA is corroborated by the spike-in data, where if you have greater than 4 cells recovered, you are able to positively identify this particular mutation. Okay. Finally, I'd like to show you a little bit of data that was obtained with a clinical research pharma company. In this case, we used a larger number of cells. They were again spiked in, but we're not plotting in the spiking number. We're actually plotting in the recovered cells in a matched enumeration sample. So a lot of the studies that we do have this kind of companion enumeration data. So we'll take 2 blood tubes, one will go to enumeration and one will go to mutation detection. So you can see here, as expected, for this larger numbers of cells, the delta CTs are significantly lower and well within the detection region. And what we're doing with these type of samples is now, after doing the analytical validation, we're moving on to patient samples. And here's an example of 2 patient samples plotted on the same graph. So here you have, on the x-axis, you have a plot of the CTCs recovered, so that's the enumeration data. And on the y-axis, you have the delta CT, and you can see that there's one patient that's within the detection zone, so positive for the G12D mutation, and another one that is just above. All right, so I think another important assay type that we wanted to validate is next-gen sequencing. It's attractive because it provides the capability to looking at a larger set of mutations, and if you don't know what mutation you're looking for, it's very useful in clinical research. Now, this data, I should point out, was a part of an R&D collaboration with Asuragen. They're a CRO based in Austin, Texas. In this case, we took 4 IsoFlux samples spiked with NDA cells again, and those were known to harbor a G13D mutation. This assay looked at 5 hotspot genes, and the expected mutation is shown here along with the measured percent mutant KRAS. So essentially, we looked at the number of cells recovered using counting, enumeration of the sample, and the matched sample went to mutational detection. And you can see that there's a really pretty good correlation between the predicted percent mutant cells and the measured percent mutant cells, and that's down to about 1%. So that's right within our sort of purity area of the samples coming off the IsoFlux separation. Of course, there's a number of other ways to look at DNA aberration. One of the more common ones is FISH, and here we're just showing feasibility of performing FISH using these samples. Essentially, we transferred the sample to a pathology slide, and we went through immunofluorescence staining and FISH evaluation. This just basically shows 2 cell images, one normal and one showing HER2 amplification. All right, so I promised I would talk about RNA next, and here's again data, just a basic analytical data that was obtained with a biopharma partner, looking at a number of different, again now, gene expression profiles for cells spiked into whole blood. So here on the left we have the standard curves. These are the cells alone, and we're looking at leukocyte markers, ALS1 and HLA-E, as well as CTC specific markers, in this case EpCAM and keratin. What this really established is, again, the detection levels for the number of spike-ins that were required in order to positively detect overexpression or just expression status of the cells. So the important experiments are here noted as experiment 1 and experiment 2, and you can see that both the red dots and blue triangles in this case, which are the CTC specific markers, show a linear dependence on the number of cells spiked in, and they're all well separated from the background, which is showed in the non-template controls. So our conclusion from this data is that when one spikes in as little as 10 cells into a tube, one can recover those and determine their expression profiles. So here's again more data from the same collaborator, and this is these are targets that are unknown to us, but essentially they are closer to real life in that some of the genes are not present, they're not expressed in the sample. For example, gene F is not expressed, and the other ones scale with, again, the number of spike-ins. So here's some other data that we've looked at using mRNA as a reporter and looking at both the extracted sample, here called cell spot, and the pre-isolation and negative fraction. The important thing here is to note the dramatic reduction in delta CT for the cell spot sample, and that's due to the enrichment of the CTCs. So both HER2 and EpCAM and cytokeratin are amplified here. CD45, the level there of the DCT remains constant because that's present in all cells. Now, if we apply a similar method to patient samples, you can see that for some samples, such as the one shown on the left here in patient 1, that had robust enumeration of CTCs, but the CT of the cell spots, so the enriched sample, was the same as the CT of the pre-isolation and in fact negative fraction, showing that HER2 is really not overexpressed here, whereas for patient 2, you see a significant difference in CT where the enriched sample shows a lower HER2 CT as compared to our controls. Therefore, there is overexpression in this patient, whereas patient 1 does not show a HER2 signature. All right, so finally I'm going to talk about one sample analysis modality rather, where we're looking at protein expression. And this work was done in collaboration with Prometheus labs, and they've developed a really sensitive method for determining both protein presence and phosphorylated protein presence in low cell number samples. So you see here what the raw data looks like. Essentially, it's a fluorescence intensity scale, and they've done analytical work down to fractions of a cell, but really above 3 cells, as you see in their graphs below the images. They have a clear path to detecting the presence of this the protein content. And this is, by the way, just published data from Kim et al. that was reported by Prometheus. Now here's some of our data that was obtained in our collaboration with Prometheus, and it compares basically a healthy control, that's the center panels, to 2 different patients that were known to present CTCs, and they again are doing evaluation across different platforms, so they have Veradex data as well as IsoFlux data. And you can see that the top panel here is labeled cocktail. That's because they've actually taken the approach of optimizing the antigen that's used to separate out samples in the IsoFlux instrument. So they're taking a proprietary mix of antibodies and conjugated those with our beads and used that to basically separate the cells out. So what does this data actually translate to once one completes the analysis? So you can see here CD45 expression counts for both patients and healthy controls. That's not unexpected. That's expressed across the board. The important data to look at is on the right panel here, and you can see that in this case, our expression of pHER1 is significantly higher when you use a cocktail, and that's higher than both EpCAM only separation and using cocktails. Now, for patient 2, you actually get recovery using both EpCAM and the Veradex approach and our cocktail of antibodies. So it's nice data in a few different ways. One, it shows the superior recovery. The other one is that it really shows that in certain situations, for certain indications, using a combination of antibodies is vastly superior to just using EpCAM alone. All right, so here's an example of data from another clinical study. In this case, we were working with Indiana University, and this is again aimed at getting both enumeration data as well as detecting mutations in this patient population. So we have a workflow that's not unusual. We obtain matched blood samples termed here sample 1 and sample 2. One of them goes to enumeration after going through the separation process. The other one goes through the same separation process and goes on to a mutational detection assay. Again, the earlier results in recovery are echoed here, where you can see that 90% of the patients tested to date in this study, that is, I believe 7 out of 8, have greater than 5 CTCs presented. Another really great partnership that we've been working on, and this, by the way, has been presented on by Bayer. This is one of 2 posters that they've presented using IsoFlux data, is looking at apheresis samples, as I talked about earlier, and really taking the same match sample and doing separation across a large number of different either existing or emerging CTC isolation platforms. And you can see some of the providers listed here, as well as the data slide down below, and I'm just going to focus on that. Again, I believe apheresis will be very important for a number of reasons. One is chiefly because it provides a lot larger numbers of CTCs. And now we have data in collaboration with Bayer, not just enumerating the samples, but we know that their downstream data that's being obtained really matches the numbers of CTCs that are being enumerated. Another very important distinction here is that we've been able to process these samples frozen on the IsoFlux, which is really of great benefit when you start to take into account the logistics of performing sample analysis at many different sites and doing that in a time-sensitive manner. So basically what the study entailed is, again, obtaining a large number of cells, essentially buffy coat directly, and then splitting that up and looking at different results of separation experiments at different sites. Here are listed 2 CellSearch sites, as well as the results using our instrument. And as you can see, as I've told you all along, the numbers of CTCs recovered are much, much higher. All right, so finally, I'd like to conclude with an invitation to join us in Boston and an invitation to adopt the IsoFlux platform for your CTC analysis experiments. I think we've really worked hard, and we're able to achieve a very efficient recovery of CTCs from samples. As I've showed you, greater than 75% of patients that are processed as a blood tube show greater than 5 CTCs, and the numbers are much higher for apheresis patients. Now, the other important distinction, and I think what makes molecular analysis much easier, is the elution volumes for our approach are very low. They're below 5 microliters, and the system is designed in such a way that it's a benchtop instrument that's easy to use, and it allows a lot of flexibility in terms of the antibody mix that's used to separate cells out. In addition, we have an in-house discovery services team. We've worked with a lot of our early collaborators, both by processing samples here, as well as sending out pre-commercialization units to a number of different pharma companies that are being used in-house in those places. So finally, I'd like to just tell you a little bit about Fluxion, and I think a big thing that we bring to the table here is the fact that we actually have a proven track record of service for the life science industry. We have developed 4 different instruments thus far. They're being used by all major pharma companies in preclinical discovery, and they all have in common a technology core of microfluidic development and cell-based assays. And with that, I'd like to tell you about the launch of this product. We are launching the product at the upcoming World CTC Summit in Boston. We'll talk there again and show some additional data, and we will be showing up the system, and we invite you to our booth, and we'll be also accepting system orders with an 8 to 12 week delivery time. We have a very exciting early access program that we'll be enrolling our first set of customers in, and if you want more details about that, you can contact Michael. Again, if you'd like to have processed samples by Fluxion, we have a full range of analytical services that can perform both post-isolation and downstream analysis of the CTC samples. And we've done a number of those with our existing partners. With that, I will conclude. Michael Schwartz, another one of the panelists, is the best contact for you if you'd like to learn more about the technology and our product availability. And I thank you for your time and look forward to your questions. Okay, so I've just been handed a list of questions here. So there's one question on the total daily throughput. We can, with one instrument, you can run 4 samples at a time. We estimate you can do about 4 of those runs a day, so that's 16 samples a day. Of course, that's assuming that you have that many samples available to process. The other question was, can we recover single cells from the sample? So the current design does not provide single cells as an output. We provide a combination of circulating tumor cells and white blood cell background. Now, it does enable some of the cell picking technologies that are out there by providing a pre-enrichment step. Again, you can use a pipette or some other single cell manipulation device to get single cells out. The other question was, how long are blood samples stable for? Again, typically what we do is we use a blood draw that goes into an EDTA tube and is stable for at least 36 hours. So we process blood samples within 36 hours. However, we have, as I pointed out, processed bank samples that were derived from apheresis extractions. So those were frozen and stable for much longer. Okay. There was one other question that's around the sterility of the cells. Are the cells being kept sterile? The answer is yes. I mean, our devices are sterile, but the sterility of the outbound sample is dependent on a good sterile technique when the samples are loaded. If you noticed, looking at the cartridge, we actually have a membrane that goes on top of the sample walls. That's meant to help preserve sterility of the sample. And we have actually done with collaborators a number of cell culture studies, and we've been able to put in ex vivo primary cells and pull them back out of the blood tube using the IsoFlux, and then perform cell culture. So it is that basically validates the fact that it's possible to keep the sample sterile. There was another question on the use of custom antibodies. The answer there is that we basically have two types of kits that ship with the instrument. One has pre-conjugated EpCAM antibody. The other one just has IgG conjugated beads. So it's mouse immunoglobulin to which the user can add one or more antibodies directly to either the sample or the beads before separation. All right. I'm sure I think there are other questions that I haven't gotten to. And I don't know. I think we're just about out of time. So I believe that we're just going to address those offline. So rest assured, we will follow up with the answers. Thanks again for your time, and I hope this was interesting, and look forward to talking to you in person in Boston if you'll be attending the conference.